Hey guys, welcome to Unit 4, Young-Splaining Cell Communication. This is going to be a quick overview. Well, maybe not so quick. Y'all know how I get to talking. It's going to be an overview of Unit 4's Section 5.6, which covers cell communication. So first things first, you need to make sure you know and understand the three stages of cell communication, which are reception, transduction, and response. How a receptor protein can recognize signal molecules and start transduction. How a cell signal is amplified by a phosphorylation cascade, an example of a second messenger and its role in the signal transduction pathway. How a cell responds to the nucleus turns on genes, whereas if it's in the cytoplasm, it can actually activate enzymes, and why apoptosis is important to normal functioning of multicellular organisms. So, there are two types of cell signaling. Cell signaling can either be local or long distance. And if it's local in animal and plant cells, it can either be done through direct contact. Now, the direct contact that allows things to pass from cytoplasm to cytoplasm in animal cells is called a gap junction. And if it's in a plant cell, it's called plasmodesmata. Animal cells also are able to communicate through cell-cell recognition, as you can see here, where a cell has receptors on their in their membrane, and then they're able to recognize a neighboring cell and then pass that signal through that direct cell-cell recognition. Now, direct contact does not use any type of chemical messengers. And when it comes to plant cells, the most understood type of local signaling has to do with the plasmodesmata. We don't know anything else about how plants would communicate cell-cell locally. So if you are involving chemical signals or ligands, then you have three different types of cell communication. You have two local and one long distance. The first local is paracrine signaling, and paracrine signaling is going to use things such as growth factors, where you're going to have a local regulator that has secretory vesicles in it, and those secretory vesicles are going to actually release the growth factors into the area around it, and only the target cells will be able to actually respond to those growth factors. So just because a cell is in the area does not mean that it would trigger a response. Remember that in order for a cell to actually trigger a cell communication pathway or have a cellular response, it must contain the membrane receptor that the ligand is able to bind to. Another type of local communication is synaptic, and synaptic communication is going to use those neurotransmitters. So it's going to send electrical signals, and this is what we see in our nervous system. And last, the type of long distance communication is endocrine signaling. Endocrine signaling is going to use your hormones. So you're going to have hormones that will actually travel through the bloodstream throughout the body. And with endocrine signaling, again, only the target cells will be able to bind to the specific hormones. So when we talk about paracrine signaling, it's important to know that in many cases, animal cells are going to communicate using a secreted messenger, and these molecules are going to be able to travel only a short distance. So that's where we have things like our growth factors. Growth factors will actually stimulate cells to grow and divide and to perform other type of cellular activities. And so this type of local signaling in animals is called paracrine signaling. Synaptic signaling also occurs in animals, and it uses the nervous system, and it's going to use these neurotransmitters, which will be released in response to electrical signals. The local signaling in plants, again, is not well understood beyond our knowledge of the plasmodesmata, which does not use any type of chemical signals. This is a direct type of contact, cell-cell direct contact. Now, long distance signaling, again, is going to be your endocrine signaling, and endocrine signaling is going to use your hormones. These chemical hormones are actually going to travel through the bloodstream, through the circulatory system, and then it will reach its target cells. Remember, the ability of a cell to respond is strictly dependent upon whether or not that cell has a specific receptor for the signal that has been released. When we think about cell communication, there are three um, steps to cell communication. And we understand this due to the work of Earl W. Sutherland, who actually won a Nobel Prize for his work on the hormone epinephrine and how it acts on cells. We know, thanks to Mr. Sutherland's work, that the three steps are reception, transduction, and response. So in reception, you're going to have a signaling molecule, which is a ligand, which is going to be a chemical signal that will bind to a receptor. Now, receptors can either be membrane receptors or they can be intracellular receptors. Membrane receptors are embedded in the plasma membrane and will bind signals that are typically water soluble, hydrophilic. Remember, anything that is hydrophilic will not be able to cross the plasma membrane. So these signals can't cross the membrane, so they will bind to a receptor that is embedded in the membrane. Now, any small nonpolar signals, such as steroid or thyroid hormones, will be able to actually cross the plasma membrane and bind to intracellular receptors, and we'll look at that a little bit later. But for now, let's just look at the three stages using a membrane protein and a signaling molecule that is hydrophilic or water soluble. So the signaling molecule binds to the receptor, and this is your point of reception. Now, typically, whenever a signaling molecule binds to a receptor, there will be some type of conformational change in shape, and this will be your first act of transduction. Now, this receptor molecule is going to actually pass the signal along the relay molecules until ultimately you end up with a response when it reaches its target. The target can be something as far as a transcription factor, which will turn on and off a gene, or it can be signaling the cell to divide. It could also be something as far as controlling enzyme activity, turning enzymes on and off. The transduction process is usually multi-step, and that actually helps to control the cellular response when you can turn it on and off or up and down in several different areas. Okay, so again, this is reception. And when we're talking about membrane receptors, there are three main types. You have your G protein coupled receptors, which are your GPCRs, and your RTKs, which are receptor tyrosine kinases. And the simplest one would be your ligand-gated ion channel receptors. Now, GPCRs are the most common, so that's the one we spend the most time on. But let's just look at a simple GPCR diagram. Whenever you have a G protein coupled receptor, you're going to have these three membranes for sure. You're going to have your G protein coupled receptor, you're going to have a G protein, and you're going to have an enzyme. These three membranes will be attached to either the cytoplasmic side, the G protein, or embedded in the plasma membrane. Now, what happens is a signaling molecule or a ligand binds to the G protein coupled receptor, and it's called a G protein coupled receptor, meaning that it will couple with the G protein. Once activated, it will couple with the G protein, stimulating that G protein to switch from its inactive state to its active state. Now, the difference between an inactive G protein and an active G protein is the molecule, the energy molecule that it is bonded to. If it is bonded to GDP or guanosine diphosphate, the G protein is in its inactive state. Once it binds to the GPCR after it has received the signal, then the G protein will release the GDP and bond with the GTP. This activates the G protein. Once the G protein is activated, it will move through the membrane to activate an enzyme. Now, once this happens, the signaling molecule will be released. Any conformational changes in shape are temporary, okay? And that's one way that the signal can be regulated, because this G protein, once it activates the enzyme, the GTP will be dephosphorylated, turning it back into GDP, which deactivates the G protein. This G protein will only be activated again in response to another ligand binding to the receptor. Therefore, this is one way to regulate the cellular response. The G protein, again, activates the enzyme. The enzyme will either trigger a cellular response or activate a phosphorylation cascade, or activate a relay protein. There's several things that can happen with this enzyme, and it really depends on the class of enzyme, the type of enzyme, the signal that was received, and the proteins that are actually available in the cell. Every cell has a different collection of proteins, so they are going to respond differently. Even if it's the same signal, the same signaling molecule can trigger a different response in different types of cells because of the type of proteins that are available in that cell. And so our next class of membrane proteins are your RTKs, or receptor tyrosine kinases. And RTKs, the biggest difference between an RTK and a G protein coupled receptor is that an RTK can set off several cellular responses. RTKs also are energy expensive because it takes 6 ATP molecules to activate RTKs. They're the two-step process to activation with an RTK. As you see here, you have your two receptors, which are separated in the membrane. This is an alpha helix in the membrane here. And on the cytoplasmic side, you're going to have your tyrosine residues. Tyrosine, remember, is one of the 20 amino acids. Now, when a ligand binds to the two receptors, it triggers the receptors to form a dimer or come together. That's the first stage of activation. The second stage of activation involves the kinases, and remember, kinases are enzymes that phosphorylate a substrate. The kinases are going to move a phosphate from ATP to the tyrosines, and this completely activates the RTK. Now, these tyrosines, these phosphorylated tyrosines, now have the ability to activate an inactive relay protein, and depending on the protein that is activated, it could trigger a different cellular response. RTKs that are malfunctioning have also been linked to several different types of cancers. The last type of membrane protein is the simplest kind. It's your ligand-gated ion channel. This is just what it sounds like. You have an ion channel. Remember, ions, again, are hydrophilic, so they cannot simply cross the plasma membrane. They have to go through a protein to move across that membrane. In the cases of ions, sometimes you want to really control the concentration, so you put them behind a gate, basically. And so this is a ligand-gated ion channel, and so the ligand has to bind to the gate so that it can open and the ions can pass through. And then once the ligand is released, the gate closes, the ions can no longer cross. Simple as one. Now, those were all membrane receptors, but we do have intracellular receptors. Intracellular receptors are going to be receptors that are actually in the cytoplasm. Remember, anything that is small, nonpolar, can cross the plasma membrane. So typically you're going to have your steroid and your thyroid hormones that are going to, of animals that will be able to act as hydrophobic messengers that will actually cross the plasma membrane and bind to these intracellular receptors. Now, when a hormone binds to an intracellular receptor protein, we call that a hormone receptor complex. And one thing about a hormone receptor complex is that it can act as a transcription factor. So if this is a hormone receptor complex, it is able to move into the nucleus, bind to the DNA, and trigger transcription. And transcription is when DNA is copied into messenger RNA. And we know from Dr. Pepper time, remember, that DNA is transcribed to RNA. RNA is translated to proteins, and then proteins are expressed as our traits, except for when they're not. So the transcription factor has the ability to turn on a gene so that the messenger RNA can be transcribed, and once processed, it'll leave the nucleus where it will go to a ribosome, either a free ribosome or a bound ribosome, and a protein will be translated. Transduction is simply relaying the signal. And again, the signal transduction pathway usually involves multiple steps. I think I talked about this earlier. And those steps can either amplify the signal or help you to be able to better control the signal, provides more opportunities for coordination and regulation. So the signal can be turned on, it can be turned off, it can be turned up, it can be turned down, because these are multi-step pathways. Most often in a signal transduction pathway, you're going to see what's called a phosphorylation cascade. Now, a phosphorylation cascade is going to involve kinases, because remember, kinases are the enzymes that phosphorylate a substrate, but it's also going to involve phosphatases because phosphatases are the enzymes that actually dephosphorylate a kinase to put it back into its inactive state. Remember, we only want something active for as long as it needs to be. So what would happen would be that, again, this is a generic pathway. A ligand would bind to a receptor protein, which would activate a relay molecule. The relay molecule would then activate the first kinase in its phosphorylation cascade. I want you to notice that the relay molecule, although it is a protein, is not a kinase. Therefore, it does not phosphorylate that first protein kinase. The first protein kinase, once activated, is able to phosphorylate another inactive kinase. When it does that, that inactive kinase becomes activated, and then that activated kinase will phosphorylate another inactive kinase. Now, because this one was phosphorylated, so here we have the protein kinase 2, it was actually phosphorylated. This is where your first protein phosphatase will act to dephosphorylate it and put it back into its inactive state after it has activated your next protein kinase. This cascade can continue until finally a protein kinase will activate an inactive protein, and then that inactive protein will trigger a cellular response. And again, you have a protein phosphatase that will deactivate that protein to put it back into its inactive state. Now, this is only showing a 1-for-1, but when this cascade is happening, it can also amplify the signal because it can be more than one kinase that is being activated at a time. And we'll see that when we look at the epinephrine pathway in liver cells. Now, notice that previously when we looked at we talked about transduction, and when we looked at phosphorylation cascade, we had a protein, relay protein. We had kinases. We had protein phosphatases. These are all proteins that are participating in this transduction pathway, but sometimes a non-protein will get involved in transduction. When a non-protein molecule is involved in a signaling pathway, a signaling transduction pathway, we call those molecules second messengers. The first messenger is always the ligand. Second messengers are going to be small, non-protein, water-soluble molecules or ions that spread throughout the cell, throughout a cell by diffusion. Now, your second messengers are going to participate in pathways initiated by GPCRs and RTKs. And your two most common second messengers are going to be your cyclic AMP and your calcium ions. So let's look at how cyclic AMP is actually made. Cyclic AMP, the AMP stands for adenosine monophosphate. So that means that it's basically it's an RNA nucleotide, or you can think of it as an ATP with two less phosphate groups. Remember that ATP is in the class of nucleic acids because it does have the nitrogenous base, the pentose sugar, in this case is ribose, in this case the base is adenine, and the phosphate group. What makes ATP unique is that it has two additional phosphate groups held together with high energy bonds. Makes it able to store energy for cellular use. So what happens with ATP, an enzyme adenylyl cyclase will catalyze the removal of two phosphate groups from ATP, and that first phosphate group will form a bond back to the number 3 carbon on that ribose sugar. If you notice here, let's count our carbons. We have 1, 2, 3, 4, 5. This is a pentose or a 5-carbon sugar. The phosphate group is attached to the number 5 carbon, but then after adenylyl cyclase catalyzes ATP becoming cyclic AMP, notice that this oxygen on the first phosphate group will form a chemical bond back to that ribose sugar. So now it is bonded in more than one location. So that's why it's called cyclic AMP. Cyclic AMP can be turned back into regular AMP by using another enzyme, catalyzed by another enzyme, phosphodiesterase. AMP, again, is just an RNA nucleotide with adenine as the nitrogenous base. So let's look at cyclic AMP as the second messenger. Of course, this is a generic one. We're going to look at some actual concentrations and what the actual phosphorylation cascade would be and the cellular response right after this. But this is using epinephrine as a signaling molecule. Remember, this is what Mr. Sutherland actually did his work on and won his Nobel Prize for it. That helped us come up with the 3 stages of cell communication. So epinephrine will bind to, in this case, we're using the GPCR, G protein coupled receptor. Epinephrine binds to the GPCR. The GPCR will then activate G protein because it's a coupled receptor. The G protein becomes activated and it activates the enzyme. And this enzyme is adenylyl cyclase. Adenylyl cyclase, remember, catalyzes the reaction of ATP conversion into cyclic AMP. Cyclic AMP will most often activate protein kinase A. This is not a generic name. This is the kinase, protein kinase A. Then protein kinase A will typically activate either another kinase or a relay protein, which will then ultimately lead to cellular response. Remember, this can be multi-step through here. So let's actually look at the epinephrine pathway in the liver cells. So epinephrine will bind to a G protein coupled receptor, and that's only one molecule that's binding. Remember that receptor couples with the G protein, then inactive G protein becomes activated G protein, and look, we have 100 molecules of activated G proteins. These activated G proteins will then activate 100 molecules of adenylyl cyclase, and these 100 adenylyl cyclase will then will catalyze the conversion of ATP to cyclic AMP. And again, look at this, we're getting an exponential increase, okay? So here we went from 100 to here where we have what? About 10,000? Yeah. 10 to the 4th, that's 10,000, right? Don't quote me on that. Wait, 10, yeah, I think so. All right, it's not 1,000, because 1,000 will be 3 zeros. So yeah, 10,000. Anyway, cyclic AMP will then activate, as we talked about, protein kinase A. So it's going to activate 10,000 protein kinase A's. Now these protein kinase A's are going to activate phosphorylase kinase. And so then we go from having 10,000 molecules to 100,000 molecules. If you notice, this signal is amplifying. Going through this transduction pathway, this signal is getting larger and larger. And so now we have your 100,000 activated phosphorylase kinases, and then those phosphorylase kinases are going to actually activate your glycogen phosphorylase. Now, an activated glycogen phosphorylase, its job is to trigger the breakdown of glycogen into glucose 1-phosphate, which is the response. Why do we want to do that? Because glucose 1-phosphate can be used in cellular respiration to make ATP. Remember, glycogen is the way that animals store glucose molecules, short-term storage of glucose molecules in the liver and in your muscle cells, so that it can be converted to glucose 1-phosphate, and then glucose till it can actually undergo cellular respiration later on. Now, again, your cyclic AMP pathways are usually going to be triggered by RTKs or GPCRs. They're usually going to activate protein kinase A, which phosphorylates various other proteins. And further regulation of cell metabolism provided by G protein systems can actually inhibit adenylyl cyclase. One of the things that we talked about in class, remember we talked about cholera and how that bacteria will actually prevent the G protein from being deactivated, and that G protein continually activates adenylyl cyclase, which then continually converts ATP to cyclic AMP. And if it is in the intestinal cells, it's going to actually, the response will be to the release of sodium. Well, unfortunately, or fortunately, depending if it's working right, where sodium goes, water follows. And so that leads to the diarrhea and the dehydration, and if not treated, ultimately the death, all because the bacteria is able to prevent that G protein from becoming inactivated. And remember, one of the ways that we regulate the cell system is that once the G protein activates the enzyme, the G protein that is bound to the GTP, that GTP should be dephosphorylated, which puts the G protein back into an inactive state. That's a form of regulation. Our second messenger would be our calcium ions. Calcium ions act as second messengers in many pathways. because if you change the concentration of the calcium ions in the cytosol, it's going to trigger a lot of cellular responses. Now, if we look at a typical cell, what we have here is showing our calcium ion concentration. The peach color is a low calcium ion concentration, and the blue is a high calcium ion concentration. So what you have is typically outside of the cell, and in the ER membrane and in the matrix of the mitochondria, you're going to have higher concentrations of calcium ions than you do in the actual cytosol. And so this is where your ligand-gated ion channels will come into play, because not only are you going to have a calcium pump that is going to keep those molecules moving against the concentration gradient from low to high, but in the event that they are needed in the cytoplasm, then they can diffuse back across the cell through facilitated diffusion using a ligand-gated ion channel, which will only open in response to a ligand. So here we're going to look at another G protein coupled receptor being used, but this time we're going to use a chemical messenger that would require calcium ions as a second messenger. Now remember, your ligand is always your first messenger. So ligand binds to G protein coupled receptor. G protein coupled receptor activates G protein. G protein activates enzyme. In this case, this enzyme is phospholipase C. Phospholipase C's job is to cleave IP3 or inositol triphosphate from PIP2. PIP2 is a combination of diacylglycerol and inositol triphosphate. You can call them DAG and IP3. That's fine. And so phospholipase C's job is to catalyze the release of IP3. IP3 then serves as your second messenger, which will actually bind to the ligand-gated ion channel, releasing the calcium ions into the cytoplasm. Your calcium ions are the second messenger. Please remember that there's only your primary messenger, your first messenger, and your second messenger. These are called second messengers because they are non-protein molecules that actually relay the signal from inside the cell through the cytoplasm. So the signal has to already be inside of the cell. Signals inside of the cell here. They can relay it across the cytoplasm. Once the calcium ions are released as second messengers, they activate various proteins, which leads to various cellular responses. And of course, we keep saying cellular response, and the response is simply carrying out the instructions. Response may occur in the cytoplasm or in the nucleus. And if it occurs in the nucleus, it can either turn genes on or off. An activated molecule in a signaling pathway can actually serve as a transcription factor. We saw that earlier with the hormone receptor complex with the intracellular receptors, but it doesn't have to be an intracellular receptor. With the hormone receptor complex, and many signaling pathways are going to regulate the synthesis of enzymes or other proteins, and that's usually what we have in the cytoplasm. So the location of the response is important as well. If the response is in the nucleus, it is going to be regulating protein synthesis. It's going to be regulating transcription. Okay, genes will be turned on and off. If the response is in the cytoplasm, it could be regulating enzymes. It could be stimulating the cell, telling the cell it's time to undergo cell division. It could be regulating the production of proteins. Okay, so this is an example of a regulation where you have your activated protein serving as a transcription factor. So again, you have a growth factor which binds to a receptor. And if it's a growth factor, remember this will be your paracrine signaling, because paracrine is that local signaling. So the growth factor binds to the receptor, which sets off a phosphorylation cascade. The phosphorylation cascade ultimately results in the activation of a transcription factor, and the transcription factor can then turn on the gene for transcription to messenger RNA. Messenger RNA is processed, leaves the nucleus, goes to the ribosome, where it can be translated into a protein. So other pathways regulate the activity of enzymes rather than their synthesis. a signal could cause the opening of closing or an ion channel. We saw that with the IP3 releasing the calcium ions, the second messenger. IP3 is actually opening and closing that ion channel. You can also have a change in the cell's metabolism as a result of the signal. So it's not always going to be just regulating activity of enzymes, turning enzymes on and off. Sometimes it even controls, like I said, if it's in the nucleus and it serves as a transcription factor, it's going to actually control whether or not that enzyme is going to be transcribed or not. For instance, people who are lactose intolerant no longer transcribe the gene for lactase, which is the enzyme that breaks down that milk sugar. You still have the gene is still present in your genome. It's just that it is not being turned on by a transcription factor in response to the presence of lactose in your system. There are 4 ways that ACE response can be regulated, and I call this the SEAT, S-E-A-T. The S is for specificity. Remember, if a cell does not have the correct receptor, it cannot respond to the signal, cannot bind the ligand. Efficiency is response that is enhanced by a protein such as a scaffolding protein. And the way scaffolding proteins work is that it can bind several different enzymes or kinases that are going to be that need to be activated in a cellular response. And instead of having to activate the different enzymes, the actual scaffolding protein itself is activated, which activates in turn the other molecules, and that's more efficient because you're getting it done faster with less work. Amplification we talked about, where you can have that phosphorylation cascade, and amplification is simply the signal getting bigger, and of course termination of the signal. You don't need the cell continually triggering a response off of one signal, so the signals have to be terminated. So think of it as far as when the ligand concentration decreases, then your cell's response is going to also start to decrease, because if there's no ligands to bind to the receptors will go back to their inactive form. Remember, those changes in conformational shape are temporary. Okay. Also, the response is also dependent on the type of cell. Same signal can trigger a different response depending on the type of cell, the proteins available in the cell, or even the receptor that it binds to in the same cell. So these are different cells, and in one cell you get this response, and in the other cell, same ligand, same chemical signal, you get a different response. You can also have what's known as crosstalk, where a different ligand can bind to a different receptor and actually activate or inhibit a pathway. So if we look at it, this particular ligand is triggering a response here. This ligand, if this ligand didn't bind, this other ligand could bind to this receptor, and it could crosstalk to also trigger this response by activating this enzyme, or it could inhibit this response. Even though the ligand has bind to tell this response to occur, this one could actually inactivate the enzyme, which would inhibit the response. And the other thing, like I said earlier, depending on the type of receptor, could also change the response. So if you have a different receptor, it could lead to a different response. This is what I was telling you guys earlier about the scaffolding proteins and efficiency. You can see the scaffolding protein here, where there's 3 different protein kinases here. All of these are involved in that cell signaling pathway. And instead of having to activate these 3 different kinases, instead the receptor protein will actually activate the scaffolding protein, which will in turn activate all 3 of these. That is a more efficient way of cell communication. We talked about amplification and termination already. And the last thing is apoptosis, or programmed cell death. Now, there are more than one way that a cell could die, but the most understood is the apoptosis pathway. That's the most understood programmed cell death type. And so what happens with apoptosis, the components of the cell are going to be chopped up and packaged into vesicles that are digested by scavenger cells. The reason this is done is to prevent the dying or damaged cell from actually damaging the neighboring cells, because remember, there are digestive enzymes and other types of enzymes inside of that cell that do not need to leak out. In humans and other mammals, several different pathways, including about 15 caspases, can carry out apoptosis. In addition to caspases, you also have proteases, nucleases, lipases. Remember, your proteases are going to digest the proteins, the nucleases are going to break down the nucleus, and lipases will break down your lipids. Apoptosis can be triggered by signals from outside of the cell as well as inside of the cell. And the internal signals typically are going to result from irreplaceable DNA damage, excessive protein folding, or remember we talked about the Hayflick limit, where the cell can no longer actively divide without actually damaging the DNA. Let's see. For example, apoptosis is part of normal development for hands and feet in humans, paws, and other mammals. It is also could be involved in some diseases such as Parkinson's and Alzheimer's disease, where apoptosis can also, and we also believe that apoptosis can contribute to some types of cancers. All right, so this is just showing your digits developing. Yep, and that's that. Remember, reception, transduction, response. Toodles.